Okay, so we're still working on Chapter 9, Population Genetics, but we're going to go into a little more depth in how the Hardy-Weinberg principle works, talk a little bit about selection and allele frequency, and about some other factors that commonly change allele frequency. Okay, and so this is really kind of the marriage of genetics and Darwinian evolution, where genetic changes in a population of organisms are usually measured as a change in the frequency of alleles, which genes are common. The fitness, remember, is the ability of an individual to contribute to succeeding generations. And here we're kind of getting at that key thing, the variation. And variation is the presence of one or more allele of differences in traits, physical traits, and that's proteins really. And so the variation in DNA sequence causes change in proteins, which causes a difference in a physical trait. The Hardy-Weinberg equilibrium is a mathematical expression that says the proportions of genes will not change in a population if there is no mutation, no migration, as long as the population is mating randomly and large and no selection occurs. And so if none of those things are working, this relationship should be true. And it's called an equilibrium, so it can be written as an equation. And this can be used to calculate allele frequencies. And so in the case we show you, it's for two alleles, P and Q. And they're the only two alleles for this trait in the population. So the frequency of P plus Q equals 1. And I'm going to show you an example for color coat in cats. And here P is the black color coat and Q is the white color coat. P is dominant. Okay, and so you have two alleles in the population, P plus Q. And so genotypes, individuals will have two alleles, and they can be two P's, a P and a Q, or two Q's. And so in the whole population, those alleles have to equal, those genotypes frequencies have to equal 1. And so then there's a relationship for genotype frequency. P squared plus 2PQ plus Q squared equals 1. Okay, and so if we're just doing this for cats, P squared equals having two alleles of the dominant gene, BB. The heterozygote 2PQ is the frequency of the heterozygote, B, big B, little b. And Q squared is having two being homozygous recessive. Okay, and so the genotype frequency has to equal 1 because the whole population equals 1. And so let's look at what this would look like in a population. Okay, if in a population the heterozygotes and the homozygous dominants will appear the same. So P squared plus 2PQ in this case equals 0.84, and Q squared is the frequency of the recessive genes, and in this case Q squared equals 16% or 0.16. And so we can take the square root of that, and then we'll know the frequency of the recessive allele. Okay, and so that frequency is 0.4, and so it shows you there. And then if we would expect then what we get with the other part of the population, if this allele frequency is true, if it's in Hardy-Weinberg equilibrium, is that we know because the frequency of B is 0.4, that the frequency of large B must be 0.6. Okay, and so because P plus Q equals 1, and if Q equals 0.4, then P must equal 0.6. And so this population looks like it's in Hardy-Weinberg equilibrium. Okay, and so in the second generation, we would expect the same thing. Okay, and so we can here do a chart to show us And so this looks at generation Q, and so again you get the same kind of frequency there, P plus Q, zero point 0.6, zero point 0.4, and you make the chart and you do the thing and you get the same thing. So if all five assumptions for equilibrium are true, allele frequencies and genotype frequencies do not change from one generation to the next. In reality, most populations don't meet all five assumptions. And so you can tell this because you look at um, the frequency of these genes and you find out that they're not in equilibrium, that there's some change going on. And so then you have to kind of suggest what it is. And there are some things that commonly cause these changes. And one of the most common things is that there's a small population. And genetic drift happens in small populations. And two particular things are founder effects and genetic bottlenecks. And so, first, genetic drift is what it is that um, there are random changes that occur because the population is small. And so, just by chance, these things happen, um, and chance alone. Okay, and so if a population becomes established from a small population, like on an island, becomes established from a larger population, very often one allele just disappears because the founder effect. And so this can be the founder effect. It can also be the bottleneck effect, where you get genetic drift leading to the loss of alleles, in that the population, which once was large, becomes much smaller. Um, and then this smaller population is subject to random changes. And so we get that. I have a simulation embedded in the PowerPoint. I think you can play that. But but this is really common, and so you see this in human populations that are isolated. You see it um, really in endangered species a lot. That one of the big problems endangered species face is genetic drift. That because of these bottlenecks, the individuals within the population are likely to be much more likely to be homozygous at a lot of different genes, and so they wind up with genetic problems and less variation. The other thing that happens is natural selection. And natural selection is common, and we know it happens because it happens in ways that hurt us. Um, One of the most common ways it happens is that we introduce pesticides and resistance develops. And so resistance alleles are often present in the population. They're just by chance. And so here's a pesticide uh, up in the top A picture. It shows the pesticide working to get inside through a cell at a specific um, protein pore or receptor. And so it's the target site for the pesticide. And often the resistance genes are just something like having fewer of the receptors. And so by doing that, you need to get a much higher dosage of the pesticide to have effect. The cell is still susceptible to the pesticide, but it takes a lot higher dose to do this. And so this is an example of where the gene for fewer receptors just becomes favored suddenly. And once that it becomes favored, it will become favored more and more and more. So selection can also, we shouldn't talk about selection may uh, happen because it can be somewhat different than that simple case, in that often several traits are affected by one gene, and selection um, has to operate on all those genes. And changes in the population can depend upon the genotypes that are favored, and these can produce some unusual patterns where you get disruptive selection, directional selection, or stabilizing selection. Directional selection is pretty easy to recognize in that it shifts the average of the population. But disruptive selection is a little bit harder, and stabilizing selection, that's just where you favor the average. Disruptive selection, where it acts to eliminate intermediate phenotypes, and so it selects for two extremes. And we have examples of these, um, in some cases, it's for things like beak size, where there's differences in the types of seeds that become available. And so birds either have very large beaks or very small beaks. And having a very large beak lets you handle the large seeds efficiently. Having a small beak lets you handle small seeds efficiently, and intermediate beaks are just crappy at handling seeds. And so you see this phenotype uh, difference where there's lots of individuals with very small beaks and lots of individuals with large beaks. And so we see these in certain natural populations. We've recently been able to use DNA evidence to study these and laboratory evidence. Um, we've worked with fruit flies to select the number of bristles on the body. These are little short hairs. They've also done some work with these with fish. Here's an example in South America and Trinidad with guppy coloration. There's two forms, um, and in this case, the one form is larger and more colorful, and the other form is small and drab. And um, the different forms are favored in different environments. The means of selection is a predator, a pike, and if in habitats where the pike is available, like downstream from waterfalls, it's bad to be large and gaudy because you're going to be eaten. But above the waterfall, where there are few or no pike, then females select the larger, more colorful males, and that becomes dominant. Um, and so they've been able to reproduce this in the lab, and you can, very quickly you can change the frequency of the genes, and so can see that happening. Okay, and then we also get selections for averages, and even sometimes when there is directional selection, that doesn't work because if you get to the limits of what it can do, and so like for some birds, they just can't produce um, more eggs, even though if there's more food available, because they can't make enough shell. Um, in some cases, like humans have been selecting for faster horses or faster greyhounds, and after a while, it just doesn't work because there's just not enough variation to produce that extra speed, and so it may be that there's no way to do it. Okay, so we'll talk about solving problems, some Hardy Weinberg problems next, and we'll practice that in the lab.